The metabolic pathway of photosynthesis serves to convert water and carbon dioxide to sugars using light energy. There's two main parts of photosynthesis, the light-dependent reaction and the light-independent reactions. Now first, the light-dependent reactions take place in the inner membrane space of the thylakoids of chloroplasts. There's two photosystems, photosystem 1 and 2. The first part of the photosynthesis pathway actually occurs in photosystem 2. This system contains two special chlorophyll A molecules that absorb energy from photons of light. This energy then excites the electrons in the chlorophyll, releasing two electrons, which is a process called photoactivation. Now, because the electrons require photons to excite, the reaction is light-dependent. These newly released electrons will reduce plastoquinone to become reduced PQ, which is the primary electron acceptor. Now, because there's two special chlorophyll molecules, each releasing two electrons, a total of 2 PQ are reduced in photosystem 2. These electron acceptors are able to move within the confines of the thylakoid membrane due to their hydrophobic nature. Next, photolysis of water occurs. Because 4 electrons were lost in the photoactivation of photosystem 2, the reaction center has to return to its original composition before a new cycle can begin. Now, because of this, the oxidized chlorophyll, not the photons themselves, will lysis or break apart water to gain its electrons. Photolysis is the process responsible for generating electrons for the light-dependent reactions. It does this by breaking water into 2H+ for the reduction of NADP, 2 electrons for photosystem 2 replacement, and 1/2 of an O2 is waste. Now next, these excited electrons from photosystem 2 are used to generate a proton gradient. The protons that were released in photolysis are pumped across the thylakoid membrane into the lumen, or the thylakoid space. Plastoquinone transfers high-energy electrons to a series of carriers, which then pass them along amongst each other, which lowers the energy of electrons each time, eventually bringing them to the hydrophilic electron acceptor plastocyanin. This is the electron transport chain. The energy released in these transfers is used to pump the protons. In this proton gradient, a high concentration of protons accumulates in the lumen. This is partly because thylakoids are very small, fluid-filled spaces, so concentration is built up very quickly. Now, because of the buildup of protons, chemiosmosis occurs to attempt equilibrium. The protons in the thylakoid space pass through the membrane to the stroma, where this is a low concentration. The energy released from this generates ATP. Now, with photosystem 1, photoactivation is going to occur again. Excited electrons are released by a pair of special chlorophyll A molecules that are activated by photon absorption. Now, instead of plastoquinone, the primary electron acceptor is ferredoxin. The lost electrons are then replaced by the de-energized electrons brought by plastocyanin. Ferredoxin, unlike plastoquinone, is hydrophilic and thus in the stroma, outside the thylakoid. This gets reduced, and 2 molecules of reduced ferredoxin are used to reduce NADP to NADPH by the enzyme NADP+ reductase. In the event that NADP+ runs out, excited electrons that would have reduced it return to the electron transport chain, where the released energy further pumps protons with the gradient, producing ATP. Once NADP+ concentration returns, it's able to be reduced again. Because reduction is turned on and off, it's cyclic photophosphorylation. This only involved photosystem 1. Now, once NADPH has been produced by photosystem 1, the light-independent reactions can begin. Here, a carboxylase enzyme catalyzes the carboxylation or the addition of a carbon to ribulose bisphosphate. This enzyme is called rubisco. Now first, carbon fixation in the stroma is going to occur. The enzyme adds CO2 to ribulose bisphosphate, which creates 2 molecules of glycerate 3-phosphate. NADPH and ATP, produced earlier, then reduce the glycerate 3-phosphate to triose phosphate. This is because in order to maintain that 2-to-1 ratio of hydrogen to oxygen that's disrupted by the addition of CO2, hydrogen from NADPH has to be added to it with the assistance of energy from ATP. Now, the triose phosphate produced from the reduced glycerate 3-phosphate undergoes a series of reactions to produce glucose molecules in the form of starch or other organic compounds. Now, not all triose phosphates can be used for production of glucose, so the majority of triose phosphate produced actually goes towards regenerating ribulose bisphosphate. In order for the Calvin cycle to cycle continuously, ribulose bisphosphate has to be produced as much as it's consumed, and this is done with ATP. So, for example, if 3 ribulose bisphosphates are used to produce 6 triose phosphates, then 1 triose phosphate will go towards creating glucose. The remaining 5 triose phosphates will have a total of 15 carbons. Ribulose bisphosphate contains 5 carbons, so 5 triose phosphates, phosphorylated by 3 ATP, are able to produce 3 ribulose bisphosphate. And this is a process called regeneration.